My name is Grace Armstrong, and I'm a chemist with the U.S. Geological Survey Mercury Research Lab located in Madison, Wisconsin. Today, I will be telling you about one of our many Great Lakes projects titled Examining Patterns of Mercury Bioaccumulation within Lake Huron. So this project was done in collaboration with many different organizations and partners, which I will be highlighting throughout the slides. To start off, why do we care about mercury in the Great Lakes? Mercury is a trace metal and persistent contaminant, and it's harmful to both human and wildlife health due to its potent neurotoxicity. Methylmercury is a bioaccumulative form of mercury, and it's at least partially responsible for about 80% of fish consumption advisories across the United States. In the Great Lakes region, mercury is listed as one of the top 8 chemicals of mutual concern, according to the Great Lakes Water Quality Agreement. Overall, we know that there's good scientific basis that climate change is affecting global mercury cycling due to effects from drought, flooding, warming, and also permafrost melting. However, we lack any detailed understanding for the Great Lakes region itself. So, there's a general need to improve our abilities to relate fish mercury levels through model improvements, which we could do by establishing more detailed information of mercury releases to air, land, and water, as well as by using new tools such as mercury stable isotopes, which can provide previously unattainable information on both mercury sources as well as pathways within the environment. In the Great Lakes in particular, methylmercury bioaccumulation is highly efficient. So, what I mean by that is that the Great Lakes have some of the lowest water and sediment methylmercury concentrations in the world, so about 6 picograms per liter, compared to global oceans which have about 17 picograms per liter, and inland lakes and streams which range from about 50 to 500. So again, some of the lowest water and sediment concentrations in the world, but one of the highest biomagnification rates, as well as some of the highest concentrations within edible fish. So it's especially important that we understand what drives bioaccumulation within the Great Lakes region. Specifically, we can look at fish concentrations across the 5 Great Lakes. So these are fish mercury concentrations from lake trout and walleye collected by the Great Lakes Fish Monitoring and Surveillance Program. I don't show it in this presentation, but Lake Superior and Lake Huron have some of the lowest sediment concentrations of mercury in the Great Lakes. However, in this plot, we can see that these two lakes exhibit some of the highest total mercury concentrations within the Great Lakes area, some of which are above the safe consumption criteria for both state levels as well as federal recommendations. And overall, the differences in these fish concentrations are largely attributed to differences in mercury source portfolios, which I will not be going into depth within this presentation. Not only are there spatial differences in mercury accumulation across the Great Lakes region, but there's also a temporal component that's really important to consider, because the Great Lakes get the majority of their mercury from atmospheric deposition, which we know has changed over time. So looking again at the EPA Lake Trout Archive, these are lake trout concentrations of mercury over time. So starting with the lake trout concentrations in this lower panel, we can see that there's a decline in fish tissue mercury concentrations, possibly corresponding with declines in atmospheric mercury due to efforts such as switching from coal combustion to natural gas for energy. However, in Lake Huron, or this upper panel, we note that the fish concentrations remain relatively constant over time. So why is Lake Huron not responding to these atmospheric declines like Lake Ontario is? Well, in short, the mercury cycle is very complex, and mercury source exposure is driven by various co-occurring stressors within the environment, such as declines in atmospheric deposition, declines in legacy mercury pools, climate change effects such as increased storm severity, as well as increased nutrient and organic carbon input and increased invasive species occurrence, all of which can impact the mercury cycle in different ways, so it can be difficult to understand the specific drivers, making it necessary to conduct lake-specific studies. And that brings us to the main objective of this project, which is to build a robust baseline dataset of mercury and key prey items within Lake Huron using a variety of tools. And so to do that, we assessed mercury distribution in benthic biomonitors and examined the use of food web tracers to pinpoint energy pathways. We also designed specialized field efforts to examine spatial patterns of bioaccumulation within plankton, also known as cestin. Lastly, we connected lower food web items to bioaccumulation trends within prey fish, and we ultimately hope to do so for game fish. So to do that, this project leveraged key partnerships, such as with the Environmental Protection Agency for our spatial water plankton collections, as well as the University of Minnesota Duluth for our sediment collections. In addition to those larger spatial surveys, the Mercury Research Lab itself conducted targeted field campaigns focusing on areas such as Saginaw Bay, tributaries, and also air collections, which aren't going to be the focus of this talk. And with all that sample collection, we of course had a lot of sample processing. And I also really want to emphasize that developing a binational study of this scale requires a lot of collaboration, and we would not have been able to complete it successfully without the valuable inputs from our partners in both Canada as well as the United States. Jumping right into our results, we will be starting from the bottom up, first looking at the benthic pathway, or the Dreissenid muscles, also known as quagga muscles. So I'll be showing you a series of concentration gradient maps, with the lighter colors representing lower concentrations of mercury and the darker colors representing the higher concentrations. And so, in general, we noted that the concentrations within Lake Huron mussels are pretty similar to other systems, with total mercury ranging from 30 to 80 nanograms per gram and methylmercury ranging from 8 to 14 nanograms per gram. And so one of the things I want to point out with this map is that Saginaw Bay had some of the lower concentrations within the lake. And so it can be difficult to see any other trends using the concentration data alone. And so that's where carbon and nitrogen stable isotopes come in, also known as light stable isotopes. And so I will dig into these plots specifically a bit more in the next slides, but in general, these are mussels by site. And so overall, we can see at a glance that the mussel carbon nitrogen stable isotope values vary by sampling location and generally reflect diet and biogeochemistry. Starting with the nitrogen stable isotope plot, in general, nitrogen 15 reflects the trophic position and biogeochemistry where the mussels reside. So looking at the plot on the left, we can see that the mussels within Lake Huron span 3 trophic levels. And so more specifically, the mussels that live deeper, so sites like AK1 and HU45, tended to have the most enriched nitrogen 15, indicating that the mussels that are longer lived are in this area and then also are in an environment where denitrification is taking place. And overall, these mussels also have the highest total mercury as represented by the darker colors. In contrast, carbon-13 reflects the main diet source of the mussels. So if we look at Saginaw Bay again as an example, we note that mussels within the southeast portion of the bay, like SP14 and SP15, tend to have a diet that's rich in phytoplankton. However, when we look at SB5 and SB6, or the northwest portion of the bay, these mussels consumed largely suspended material in high productivity areas and had the higher total mercury concentrations. And we think that that's likely related to consuming more particles that can absorb mercury. So these are just some examples of how we can use these light stable isotopes, but we're working on further parsing apart this data. Shifting to the pelagic or the open water pathway, our water and plankton collection transects were designed to incorporate the nearshore to the offshore gradient. And so overall, these are concentration bar plots with the total mercury or the concentration on the y-axis and the transect location on the x-axis. And so each of these transects are also highlighted in the map on the right. And so looking at the total mercury water concentrations first, we could see that the total mercury in water was pretty similar between nearshore and offshore zones across the lake. And I also forgot to mention that the nearshore is marked by the darker colors and the offshore is marked by the lighter colors. In contrast, when we look at the methylmercury content within these waters, we can see that methylmercury within the nearshore parts of the transects are substantially higher when compared to the offshore regions of each transect. And so, how does this look within the lower trophic levels? So this is a plot with cestin or plankton methylmercury concentrations across the lake. And in general, the trend of nearshore collections having elevated methylmercury concentrations compared to the offshore collections is consistent, similar to the methylmercury water data. And so overall, the elevated methylmercury noted within nearshore waters and also within plankton indicate that nearshore regions may have greater methylmercury production, loading, such as from the watershed, and also increased accumulation within these zones. Switching it up a bit, given that this is a meeting geared towards fish, let's get into the fish data. So prey fish are essential dietary items for sport fish, such as lake trout, and they also integrate the benthic and pelagic pathways. We obtained samples of opportunity for this project across the lake and selected species that had the most consistent spatial coverage. This is another plot of methylmercury across the different zones within the lake. This time including different prey fish species across the lake, including gobies, which are in blue, deepwater sculpin in pink, bloater in orange, and yellow perch in yellow. And so overall, looking at these data, we noted that the benthic feeders, or the round goby and deepwater sculpin, tended to have higher methylmercury concentrations compared to the more pelagic feeding fish like bloater and yellow perch. And so overall, there's clearly differences in prey fish bioaccumulation extent, both spatially and also within the regions themselves. But why is that? Well, the answer is not simple, but in general, prey fish methylmercury concentrations are driven by fish species collected, the food source of the fish, feeding habits such as foraging depth or prey nutrient density, and also the fish length or trophic levels can be big drivers of methylmercury concentrations. So we plan to dig into this further using the carbon-nitrogen stable isotopes that I highlighted with the mussels and then also with mercury stable isotope ratios. As another sample of opportunity, we also obtained a large set of juvenile whitefish from the USGS Great Lakes Science Center. And so this is a plot of our preliminary data comparing the concentration of whole body juvenile whitefish compared to the length of each fish. And so in general, the concentrations range from 40 to 180 across lengths of 60 to 374 millimeters. And overall, we know that as the length of the fish increases, the concentration also tends to increase. And that's consistent with what we see in previous research. And so we are hoping to leverage this data to build a risk assessment model for the consumption of whitefish. An example of this type of model can be seen here, where the different consumption criterion can be seen in the colors of green, representing the EPA consumption, yellow, which is unlimited consumption, and red, which is the Great Lakes Advisory no consumption criterion. So the way that you read these plots is by comparing a point in the line with the percentage likelihood. So, for example, if we take the suckers within the Rocky Mountain Fall River, we note that suckers that are about 360 millimeters long will have a 10% likelihood of exceeding the no consumption criterion set by the Great Lakes Advisory Group in red. And so we are hoping to build a similar assessment for whitefish within Lake Huron. And you can extend that to both humans and then also to assess risk in other wildlife such as birds. To summarize the portion of this study that we discussed today, we observed that the benthic mussel methylmercury bioaccumulation can be complex, but it's consistent across Lake Huron, including in key fisheries. We also observed that with the pelagic pathway, waters and plankton are elevated in methylmercury within nearshore environments when compared to the offshore portions of the transects, indicating that greater methylmercury production, loading, and accumulation may be occurring in these nearshore zones. I also didn't discuss this a lot, but the legacy mercury within Saginaw Bay sediments doesn't necessarily equate to elevated bioaccumulation within fish tissues. And across the lake, we noted that fish mercury concentrations are dictated by location and prey items. We also intend to further explore the bioaccumulation within this study with the integration of carbon and nitrogen stable isotopes. I also wanted to touch on some of the future directions for this Lake Huron project. So specifically, as a part of our ongoing efforts, we are planning to link all of these data to PFAS. And so we actually share all of the food web samples with the EPA Office of Research Development team located in Duluth, Minnesota. And additionally, we also co-collect all of the sediments and waters. So there's a really nice potential to compare both the PFAS in this data set as well as the mercury. And then also we are analyzing all the food web items for carbon and nitrogen, both bulk and stable isotope content, as well as compound-specific amino acids, as well as with our partners at the EPA ORD. And as I also mentioned, we are building a risk assessment model for Lake Huron whitefish, and we are still working up some of that data, so we'll see how that goes. As some of the data gaps of this study, we really lacked a good spatial representation of our fish collections. And so I didn't emphasize this, but these fish that I highlighted in the study were all samples of opportunity. So we reached out to different groups, and they were able to provide us with different sets of fish in order to use them as our M members for our bioaccumulation assessment. So that was really helpful, but in general, we had a limited amount of species that were consistent across the lake, and also there weren't any fish collected from Georgian Bay, which is a prominent fishery. So we hope that with future studies, we're able to plan more in advance in order to develop more consistent surveys and also to determine drivers of bioaccumulation. And then as I mentioned, we're also continuing to to develop the base of the food web, including with both mercury stable isotopes and then also carbon and nitrogen stable isotopes, to really dig into what modulates bioaccumulation in Lake Huron. Lastly, I wanted to highlight the synergy across all of our Great Lakes projects. Ultimately, the Great Lakes are key to understanding how mercury loading to ecosystems will fluctuate in response to global initiatives to decrease mercury emissions and also with climate change. I want to especially highlight that this is just one of our projects within the Great Lakes region, and we actually conduct similar assessments across all 5 Great Lakes, with each lake getting its own unique spin as highlighted by the questions on this slide. And so these are all the points, all of the data points that we've collected across the lakes, just including our sediments in the yellow triangles and our food web and water collections in the green. And so we have a pretty vast array of samples, but you might note that Lake Michigan is empty so far. So this coming summer, we will be sampling on Lake Michigan with a similar design to Lake Huron. So again, largely focusing on the lower food web and also in areas such as Green Bay and the Fox River. And we're also hoping to maybe connect that to some prey fish, although that's not the main objective of that study. But if you have any spare fish from your collections, we would be very interested in including them as members in our similar food web study. And so similar to this study, all of our samples will also be shared with the EPA ORD team to get both that carbon nitrogen workup as well as the PFAS content. So there's some potential for some really nice data sets to be built. To conclude, I want to say many thanks to all of our wonderful partners for help with site selection and field collections, and also thank you to our funding. This is a pretty large-scale effort, so it wouldn't have been possible without binational support from all the different partners listed on this slide. I also want to highlight that most of the mercury data from this study are listed at these data releases listed here. And I'm not able to attend this conference in person. However, if you have any questions, feel free to reach out to our team at USGS Mercury Research Laboratory through our team email listed here, my email, which is listed second, or our team lead, Sarah Jansen, which is listed right here. And so thank you so much for your attention and for this opportunity to share all this exciting work.